Hi, everyone. I am going to go over chapter 10, and this is all about muscles and how they function, and the different type of muscles that we have. Let's pull it up. Right. Let's talk about the skeletal muscle characteristics. Muscles are, or most of them are attached by tendons, so tendinous insertions onto bones. The cells are going to be multinucleated. They are going to be striated, and that's these lines that you see here. They are voluntary. So when we say skeletal muscles, these are the muscles we're thinking about when we think about doing a bicep curl. We have three different types of muscles total within our body. So skeletal muscle is just one of them. Smooth muscle is another category that we have, and you notice the difference in tissue here and that we don't have those striations that we saw in skeletal muscle. They have spindle-shaped cells, and that's kind of these elongated shapes that you see. They're single nucleus to them. They are involuntary, and we are not consciously controlling these. So in places like our visceral organs, so our stomach, urinary bladder, respiratory passages, inside of some of our vasculature, we have smooth muscles that sit there that we do not control. The third group is cardiac muscle, and these are going to have striations and usually a single nucleus. They're going to be branching cells, and they're joined together by something called an intercalated disc. And these are unique to the heart muscle and allow for a more unified contraction of the muscle themselves. Of course, these are involuntary. We're not thinking about contracting our heart, and only found inside the heart itself. What makes muscle tissue different than any other muscle that we have? There are four main characteristics, and that is excitability. And excitability is simply the ability to be responsive to an electrical stimulus. They are going to have contractility, which means they are going to shorten when they are stimulated by that electrical activity. They also have extensibility, meaning that there's going to be stretch to them. And then they also are going to have recoil, and that adds to their elasticity. Important functions of our muscles. One, they are going to help for movement. They are helping move our bones or our fluids. So example, bloods, when we're looking at smooth muscle inside of the vascular wall or the tunics of the blood vessels. We use muscles to maintain our posture and our body position. Just like we talked about last chapter, muscles are going to help contribute to the overall stabilization of a joint. And of course, muscles are going to generate heat, and that's especially inside skeletal muscles as we move. They are going to generate a significant amount of heat because of friction that is caused. Also, when we are metabolizing ATP, this is going to be another area where we are going to release a lot of heat. So some additional functions that muscles can have. One is protect the organs. It is going to help form valves and do things like control our overall pupil size, so dilation and constriction of our pupil, and then also do things like cause goosebumps for us. So muscles have a lot of different functions depending on what muscle we're talking about and, of course, their location. When we are looking at skeletal muscles, and particularly when you study skeletal muscles at maybe a higher level, for now you will be studying skeletal muscles and memorizing things like origin, insertion, and action. But every single muscle has an individual artery that's going to supply it, one individual nerve that's going to supply it, and then there are a couple of veins usually that are going to have venous return from that area. So we name them or we memorize them, each muscle with origin, insertion, action, blood supply, and nerve supply. Now you won't be responsible for doing that at this point, but do know that each muscle does have a specific nerve that is going to innervate them. When we go into the nervous system, we'll kind of see how the nervous system is broken down into its peripheral parts, and each branch has a specific name, and that specific name is going to go to a specific area, including a specific muscle. What this allows us to do, and the great thing about it, is that we can then figure out when a muscle isn't functioning. If it's because of the nerve, we can trace it back to whether it's that peripheral nerve, a previous branch, or from a level at the spinal cord. We'll look at that when we get into the nervous system. Every skeletal muscle is supplied by a nerve ending that controls its activity. So that's kind of that nerve supplies that muscle. Now, nerves do supply more than one thing, but each muscle has one nerve. that is going to contribute to its electrical stimulus. Each muscle, because of its requirement for ATP, so it's highly metabolic because if we are using it, to use it, it requires ATP. That requires a vast amount of oxygen and other nutrients. Remember, oxygen, the reason we even need oxygen in the first place is because it is the major final electron acceptor when we are making mass amounts of ATP in places like the electron transport chain. When we need ATP, so for instance, contracting a muscle often over and over, we will need things like oxygen to make the ATP to make that happen. If we think back, and we'll look a little bit later in this lecture, when we study things like glycolysis or the citric acid cycle or electron transport chain, there is going to be waste that is produced by these processes. So muscles are going to also generate a large amount of waste that will need draining. So looking at the muscles themselves, if we look at the bigger muscle structure, so this top picture, it is going to be covered by epimyosin. And this is going to be dense, irregular connective tissue that's surrounding it and kind of can blend into the fascia. Fascia is going to be connective tissue that's going to encapsulate one or more muscles together. The perimyosin is going to be the fibrous connective tissue around each fascicle. So when we look at what makes up a muscle, we can break it down into individual fascicle bundles that make up that skeletal muscle. And if we look at an individual fascicle, that is made up by individual muscle fibers. As far as the coverings go, we have epimyosin, perimyosin, and then endomyosin. And endomyosin is going to be around each individual muscle fibers. Epimyosin, perimyosin, endomyosin. And we'll see these prefixes often when we look at coverings around different places inside of our body. So epi is always like on the outside, the bigger thing. I always think Epcot. This might help you study, might not. I always think epi like Epcot. And if you've ever seen pictures of Disney World, right? It's the big, huge ball. So I always think Epcot, the big outside of something. Peri is going to be sort of the in-between, and endo is going to be the inside lining of things. And so endomyosin here is going to be fine areolar connective tissue that's surrounding each individual muscle fiber on the inside. As far as skeletal attachments, we've talked previously about insertions and origination points. Origination points typically are going to be at the less movable bone, and the insertion point is going to be the movable bone. The insertion always moves towards the origination point. In most cases, there are fine details where we may co-contract and then the origination point actually ends up moving. But for all intents and purposes, insertion moves towards the origination point. Attachments can be direct or indirect. Direct meaning epimyosin fused to places like the periosteum of the bone or the perichondrium of the cartilage, so having a direct attachment point. We also can have indirect attachment points for our musculature, where the connective tissues are going to extend sort of beyond the muscle like a rope. It's called a tendon. Or sometimes it could be like sheet-like aponeurosis, so kind of like a connective of a bunch of fibers coming together. So either direct or indirect. When we look, this kind of getting into where we'll talk about contractions and how that works. So here is going to be an individual muscle fiber. So we've kind of gone down to, if we go back to this piece here, right, we have individual muscle fibers here. Back here on the individual muscle fibers, we have pieces called myofibrils, and myofibrils are going to be these units or these long pieces that we actually look at on a microscopic level and can see what's happening contractility wise. When we are studying contraction or talking about things like sliding filament theory, which is going to be the sliding or contraction of these muscular fibers, we look at the functional unit, which is going to be a sarcomere. And because of all of the overlap and the different densities we see, we get these lines created. And we can map these lines and look at these striations or overlapping of striations that we see that allows us to sort of watch and see how a contraction happens. These cells are going to be important. Glycogen storage is important around the muscle while it is not infinite, and we will run out and have to go to other places like the liver where we store glycogen. We do have some storage of glycogen inside of our muscle fibers. Now, the reason we care about this is because glycogen is basically a stored up packaged sugar that we can break down to get ATP. And since we need ATP to do a muscle contraction, it's pretty convenient that it sits in the muscle. Again, there is a limited amount, and we'll have to go to other places in the body where we're storing this to make more ATP when we are doing long activities and need a lot of ATP for muscle contraction. And of course, we need oxygen, right? Because if we are going to make any new ATP, we will need oxygen in those areas. Okay. Myofibrils, muscle cell, densely packed, and you can see this myofibril. We've kind of talked about this already, and that individual strings or strands of myofibrils together, bundled together, makes a muscle fiber. So myofibril is what we're looking at here. Okay. And then these are those contractile units. So when we just look at one piece of it here. Now there are sarcomere upon and end-to-end sarcomeres, right, that make up this long rope. And so that's kind of an important fact in that when we are doing a contraction, it's not necessarily end-to-end, but it's individual units backed up together that are sliding over each other end-to-end. These striations that I talked about, those overlaps, so we have the H zone here, and this is going to appear slighter in the midsection of the A band. So here's the A band that we see. And this is just going to be where filaments are not overlapping. When we talk about these filaments, this is going to be what we call the thin and thick filaments. We have the thin filaments, which are going to be the actin, and the thick filaments, which are going to be the myosin. We'll talk about actin and myosin filaments in just a moment. So some other lines, an M line is the protein myomesin, which bisects the H zone. Z-disc line, we have thin and thick filaments. That's what I was just talking about, the actin and the myosin. And the sarcomere, again, is just between two successive Z-discs. So we're just kind of looking Z to Z is our sarcomere. And right here, when we're talking about the sliding filament theory, and we could see the thin filament in green, and the thick filament, which is the myosin, in the purple color here. And so we're looking at sort of the overlap of this green actin and this purple thick myosin. We'll talk about what's happening when we get a contraction. So here is another look at it. So here's that green actin. This is the thin filament, what we call the thin filament. And here is the thick filament, which is the myosin. So here you could see the myosin, and here you could see the actin again up here. Some important pieces here that we have that I want to point out on this slide. So actin is going to have this troponin. See that here? And we see tropomyosin as sort of these bands going along these fibers of actin. So these are going to play important roles here. On the myosin, we have the myosin heads. And these are going to be important because this is going to be what's going to bind to the actin binding sites. Also on the myosin is going to have an ATP binding site. This is where the whole function or the purpose of ATP in a muscle contraction comes into play. This is the hinge region, and we'll talk about the myosin head and how it hinges. Here's the binding site. Notice that the tropomyosin is covering the binding site when we look at it sort of in this picture here, in this thin filament picture. That's important because that covering does not allow the myosin head to attach here yet. So this has to be unbound, and we do that when calcium is present. Calcium kind of brings me to this next slide. We have the sarcoplasmic reticulum, the SR, and this is going to be surrounding each myofibril. So you can see it's sort of encapsulating this myofibril, and this is going to help function in the regulation of the intracellular calcium levels. So it's going to help store and release the calcium. Now, if calcium is stored here and we release it from here, it needs somewhere to go. And that is going to be in these T tubules. And that's these lighter bands that you see here. We have the sarcolemma and these T tubules. This is important because this is how we're going to get calcium out of the sarcoplasmic reticulum. So here is the sliding filament theory. When we have the myosin, so before we talk about the details of what happens with calcium and everything, this is how this works. So we have the myosin that has these myosin heads. These myosin heads are going to have to bind to actin and pull it over itself and constantly unhinge, grab onto actin, pull it over, let go, unhinge, pull it over. And so actin is constantly being pulled over myosin when things like calcium and ATP are present. They're going to be two important factors to allow this to happen. Let's picture there is going to be some sort of stimulus, action potential coming from the brain, right? And we're sending that electrical impulse to the area where the nerve is going to innervate the muscle. And so we look at this place here. And so electrical impulses don't really do anything for the muscle itself. The electrical impulse at the synaptic end bulb, which is this here, there's going to be this area called a synaptic cleft. And really this is the space between the muscle where the motor end plate is at and the nerve itself. Well, the way that we are able to stimulate the muscle is going to be the release of something called a neurotransmitter. Now, this is the same thing we'll talk about that happens between nerve to nerve. So as an electrical signal is going from one nerve to the next, those nerves are not actually connected together, but have to use the synaptic cleft or this spacing between each nerve, where we'll release a chemical signal in the form of things like acetylcholine, which is a type of neurotransmitter. So we see these this nerve impulse come down to the end bulb, and it starts to release these neurotransmitters. This is the neurotransmitter kind of built up in these vesicles, and these vesicles come up to the end or the plate, and it's going to release that acetylcholine by exocytosis. So now it's in the synaptic cleft. And the neurotransmitters then can go to the motor end plate and go through and attach to a receptor, where now the muscle can be stimulated to do things like release that calcium, which is sort of the starting point of a muscle contraction. As we just saw, that muscle contraction, when that calcium is present there, we can then bind to troponin, which allows the relaxing or the unwinding of tropomyosin, which then reveals that binding site on actin, so myosin can then attach to it. So this is exactly what's happening here. And so these steps. So when that nerve reaches the axon, calcium channels open. Calcium ions enter the axon terminal. Calcium causes synaptic vesicles to release acetylcholine, diffuses across the synaptic cleft. If enough acetylcholine is released, it becomes permeable to sodium, and the cell is going to be then depolarized, and we can then have a muscle contraction happen. So that's sort of what's happening as far as the nerve into the muscle, so the transmission of that impulse. Let's see, we talked about this already with that video. So a muscle fiber contraction is going to be all or none. When myosin head attaches to the actin filament, we create this cross bridge here. We are going to release that ADP and that inorganic phosphate, initiating what we call that power stroke, and that's where that actin gets pulled across the top of the myosin filament. Now, for that head to be released, that ATP has to bind for that to allow that myosin head to go back to where it's at. Now, this is just one single bit, right? Like this is just one little pull of that actin filament. So we need to look at what happens when we have a sustained muscle contraction. So here's the stimulus. So if we look at these charts over here, we're looking at time and the amount of tension. We see contraction, and that's maximal tension of a single twitch, and then relaxation phase. What I want you to notice here is that contraction happens very quickly. The relaxation takes a little bit longer. And so we can also look at what it takes to depolarize and repolarize a cell. And the repolarization event takes longer. Well, this is good for us because if we have this stimuli, and the stimulus is that electrical impulse releasing things like acetylcholine, those neurotransmitters, if we do that closer together, that relaxation takes so long that sometimes the muscle isn't all the way relaxed yet. And so what we can do is then get a harder and more forceful contraction if we increase the spacing between the stimulus. So we don't relax all the way, we contract a little more. We start relaxing, we have another stimulus, we contract more. And so we get this, what we call an unfused or incomplete tetanus. Now we often think of tetanus as a bad word. We think of getting a tetanus shot if we get a puncture from a rusty nail. Well, that's only because what that can lead to is the continual contraction of muscles where they do not let go. But really what tetanus is a sustained muscle contraction, particularly when we have a fused tetanus. So if we look at this bottom chart and we look at stimuli being so consecutive and close together, right? So this is a summation of all of that. We see a constant contraction. So the response or the forcefulness of a contraction and the length of that contraction depends on how often we're getting a stimulus to that muscle and stimulating the release of calcium, the binding of the calcium to the troponin, tropomyosin unwinding, revealing the binding spot. The myosin head attaches to actin, pulling it over. And the more we do that over and over, we get a forceful contraction. And when we think about doing something like a bicep curl and holding that bicep curl or that, let's say if we're holding a dumbbell, holding that dumbbell in place is this, right? We're getting constant sustained stimuli that's allowing that forceful hard contraction to be sustained. So twitching, so that's just a single brief contraction. So that's just going to be a single stimulus. You see this in the top right-hand corner. This is not really a normal muscle function, right? Like we've all had little twitches before. This is what's happening. When we sum those contractions up, that is where we get those sustained muscle contractions, and you see that here. This is where the energy component comes in. You saw that ATP was really needed twice when we were talking about the theory of or the sliding filament theory, where that actin is being pulled over the myosin. We needed ATP or energy to bind, but we also needed energy or ATP after we're done with the power stroke and pulling that actin over for the myosin head to release. ATP is required yet again. And so ATP is very important for muscle contraction because without it, nothing happens. So ATP, immediate source of energy for muscle contraction. We have some ATP just stored in muscles. So we do have ATP sitting around. And this is very small amount because we use this up very quickly. After we use that ATP that's sitting around, which is not a lot, we have to find other ways to make or get ATP. The first way is direct phosphorylation. And so when we say phosphorylation, when we look at ATP, which stands for adenosine triphosphate, tri meaning 3 phosphates. So that's what we are talking about. When we do phosphorylation, we are taking that phosphate off. And when we break a phosphate bond, we get energy out of that. Okay. So if we are to do phosphorylation of ADP by creatine phosphate, so we're going to add a phosphate to this adenosine diphosphate and make it adenosine triphosphate. We can do that by creatine or phosphocreatine. And so we have a creatine with a phosphate attached to it. Now we don't have huge amounts of this in our muscles either, but we do have stores of it. And when we do things like quick contractions or the very beginnings of a fight or flight type response, we'll use the ATP sitting around, and then we might take a phosphate off of a creatine phosphate. And so this is where the idea of supplementation with creatine comes into play, and that there's this idea is that if we can replenish the amount of creatine that is there, the more creatine phosphate bonds might be available to do quick contractions for, let's say, doing a quick deadlift or a quick bicep curl. The next way that we would do this is anaerobic glycolysis. So the first ways that we do anything in the very beginning is exercise. We aren't worried necessarily, or we haven't gotten to the step of using aerobic type respiration, meaning we aren't making ATP in ways that require oxygen quite yet. So the next step would be to do glycolysis. We have glucose, and so we have that sugar. Glycogen is how we store it, right? So if let's say we eat a bunch of food and there is sugar or carbohydrates in it, we would then take that glucose that we've broken down. If we want to store it for later, we would package it as glycogen. We might store glycogen in places like the liver long term, or maybe in places like the muscle in small amounts. We take that glucose and break it down through glycolysis, and this happens out in the cytosol. So we're not quite inside of the mitochondria yet, but in the cytosol. And part of what we get is pyruvic acid. That gives us a small amount of energy, and that gives us 2 ATP. So not a lot when we do this. When there is not oxygen present, we convert that to lactic acid, which is released in the blood. When we get about buildup of lactic acid inside of our muscles, this is the burn you feel when you're exercising, is the buildup of that lactic acid. Now, this is not what makes you sore the next day. We convert this back very quickly. Soreness is really going to be the result of tearing little micro tears of the muscle itself. But the buildup of lactic acid happens when there's not oxygen present. We then can also use aerobic pathways. And so we might do this by doing aerobic respiration within the mitochondria itself. So this is breaking down glucose either from glycogen or just from glucose itself. But we do this through pyruvic acid. This is the aerobic cellular pathways that we would have. And we can use aerobic respiration from fatty acids, proteins in the form of amino acids, pyruvic acid straight from glucose. But all of these things, the intermediate will be the pyruvic acid, and then we can get ATP out of places inside the mitochondria. With that in mind, is going to be the citric acid cycle we could do this with, or we could do and get smaller amounts, or we could do this with the electron transport chain, and that's going to give us a mass amount of ATP. And so we get about 32 ATP through aerobic means. Remember, all of these were by aerobic respiration. And so about 32 molecules of ATP is our net gain. Some of these things are going to cost us a little bit of energy, by the way. So we get a net of 32 for every glucose molecule that we break down. Now, this is a slower reaction and also requires oxygen to be present. Some of the byproducts, water and carbon dioxide. This is what we utilize when we are doing sustained events or needing ATP for longer periods of time. Backing up, this is that direct phosphorylation. So we had that creatine, phosphocreatine, or creatine phosphate. This is how we can get ATP out of that, right? So we're breaking it down. About 1 ATP per creatine phosphate. This is our fastest way and kind of the first step after just using up ATP that's sitting around. After that, we might use anaerobic pathways. And then if there is oxygen present and for more long-term things, we would do these type of pathways. So aerobic pathways. So citric acid cycle or Krebs cycle, same name or same thing, different names, or the electron transport chain. Okay, so now that we talked about that, let's talk about muscle fatigue and oxygen deficit. If muscle activity is strenuous and prolonged, we end up with muscle fatigue. There is going to be different ion imbalances that we end up with, and of course, lactic acid buildup within the muscle. There's not oxygen present. We saw that when we convert pyruvic acid into lactic acid in the end stages of glycolysis if there isn't O2 present. And of course, ATP supplies will decrease. After exercise, there is an oxygen deficit repaid, and deep breathing is the way that we're doing that. So this kind of explains why we start breathing and our respiration rate increases. But it also explains sort of this thing we called EPOC, excess post-exercise oxygen deficiency. We have to replace all the oxygen that we borrow. And so this is why we still are breathing heavy even after we finish exercising, because we still need a lot of oxygen to put back oxygen where we borrowed it from and get ourselves back to normal. Let's talk about the types of muscle contractions. We have sorry where these slides were going. We have isotonic and isometric type reactions. So the isotonic reactions or contractions, these are where the myofilaments are sliding past each other. This is going to be in the form of concentric, and I think of a C coming in together, so the shortening, or eccentric, where they are slowly lengthening. Isotonic contractions, when you think about a bicep curl, bringing it up and bringing it down, both fall into isotonic. So we're changing the length of the muscle in an isotonic contraction. Isometric contraction is when the muscle is contracting, but it is not shortening or lengthening. If you were to stand up next to the wall and push against the wall, you can't move that wall, right? The wall has more force exerting back on you than you're able to put on it. So the shape or the length of the muscle is not changing. However, you are still contracting. This is an isometric type contraction. So tension of the muscle increases, but you are unable to shorten or produce any type of movement. Isotonic, movement, change in lengths. Concentric is shortening. Eccentric. I will note here, eccentric, this is when, let's say we're doing a bicep curl and we have the weight up. That was the concentric contraction. We can slowly lower that weight and we're still using the bicep to do that, but we're doing an eccentric type contraction. This is also just as an important way to train and strengthen muscles. And then isometric, we aren't going to change the length, and that's because the amount of force it would require is more than we can produce. And so while there is tension, there is not a change in length of the muscles. The force of contraction depends on the number of cross bridges attached. So between the myosin heads and the actin is what we're talking about. And this is going to be affected by the number of muscle fibers that are stimulated or the amount of recruitment that happened and the relative size of the fibers. So hypertrophy or the increase in size of the cells is going to increase the strength. This is why someone who has really big biceps, they're stronger than someone who does not have as big a biceps, right? And then, of course, frequency of the stimulation. We saw that those consecutive stimulations being so close together gave us a more forceful contraction. So this is also part of this equation. And then, of course, something else we haven't talked about is the degree of stretch. When we visualize these actin and myosin fibers, there is going to be a starting point that makes the most sense, that allows us to have the most forceful contraction. Now, picture if it was spread all the way apart. So sort of like let's show this, stop sharing. So picture the actin and myosin fibers, right? And we have this overlap of them. Actin and myosin isn't overlapped enough. Because that, there's not enough cross bridges that are happening, meaning that this contraction is going to be weak. There's also the event where there's too much overlap and we aren't able to get a forceful contraction. Somewhere in between is going to be this just right, where we have just the right amount of actin and myosin cross bridges to allow a forceful contraction. Brings us to this idea of the degree of stretch of muscle. This also accounts for why, let's say, sometimes when you're in the gym, if you start at certain angles, some of the contraction already starting allows you to pull that weight easier because of the overlap. Now, also, this does have to do with physics and the angles, and certain angles are going to be easier, but that's more of a biomechanics type lecture. Force of contraction, large number of muscle fibers created. The bigger those muscle fibers actually are, the more close together or the summation of the events and tetany happening is going to increase. And then the sarcomere stretched in slightly over 100% of resting length, so the length of it, right? So all of these things are going to contribute to more cross bridges and more contractile force that's able to happen. Velocity and duration of contraction influenced by the fiber type, the load, and of course the recruitment that we have. So how we do in the gym, right? If we're lifting a small amount of weight versus a big amount of weight. When we talk about the type of fibers, whether we are talking about fast glycolytic or slow oxidative fibers, when we look at the different types of fibers that we are going to have two bigger groups. So we have slow twitch and fast twitch fibers. Slow twitch fibers are going to be oxidative, meaning that they're fatigue resistant, meaning that they are going to be used in events where we're doing more long-term things where oxygen is present. The fast twitch ones are more fast things, and oxygen doesn't need to be present. Now, the difference of these fibers is that these fast twitch ones become fatigued very easily. The slow twitch ones can last a little longer and don't become fatigued. So depending on which one is going to give us either more velocity or more duration, right? And so this comes to the idea that when we are in the gym and let's say we are doing a lower weight in the gym, we may do more reps of it, right? And we do more of an aerobic type training with it. So let's say we're doing bicep curls, but we use a 5-pound weight. We might be able to do reps of 12, 15, 16, as opposed to if I gave somebody a 30-pound weight, we might do a 3-set max or a 4-set rep with that. And that kind of explains this use of either fast glycolytic or fatigable fibers or these slow oxidative fatigue resistant fibers. So adaptations to exercise. So endurance exercise leads to increased muscle capillaries, so more blood flow there, the number of mitochondria and myoglobulin synthesis. This is why you can start training for a marathon, and at the beginning you were out of breath and can't do it, but then can build up to doing it. So this is endurance type exercise, and our body tends to adapt. So the greater the endurance, strength, and resistance to fatigue if we are exercising. And if we are training for a marathon, we might even change the fibers that we have inside of our muscle to fast glycolytic into fast oxidative type fibers, meaning that we are going to be able to utilize oxygen more efficiently and do things for longer events. So just kind of shows us that our muscles are adaptable, but we also can build up strength in muscles when we are lifting heavier. And that's what's required to increase gains as far as like our PRs or being able to do a 1 max rep. That's our resistance exercise. So usually resistance exercise, because of the nature of those fibers, are anaerobic type exercises, and is going to lead to what we want, which is bigger muscles or muscle hypertrophy. With those, we are going to increase the mitochondria, myofilaments, glycogen stores, and the connective tissue around them, and gives us, this appearance of bigger muscles because, well, they've gotten bigger. That is it for muscles. Hopefully that answered some questions or helped explain some of these topics. Happy studying.